Now it's time for the science. I'm going to turn it over to our moderator, Dr. Tom Friedman, who has a big job. He's chief of the Laboratory of Molecular Genetics in the Division of Intramural Research at NIDCD. And he's going to begin by giving us an introduction to genetics, which is a complicated subject, so he's going to give us a frame for understanding the subsequent talks. And then 3 other scientists will describe their own research. You can find bios on the scientists in your program, but there have been a couple of changes, or there's been one change in the program. Unfortunately, Dr. Richard Smith is unable to be with us today, and so Dr. Ella Aziez will be, his collaborator, will be speaking to you. Another change, due to the weather, Dr. Zhenyi Chen from Boston was not able to get out of Boston to Rochester last night, and so he'll be joining us by Skype. We'll have our format will be first Dr. Friedman, then the other 3 scientists, then we'll have questions from the audience. So as they're talking, think about what you'd like to know. Thank you very much for coming to Rochester, and Dr. Friedman.
So thanks so much for inviting me. It's a real pleasure and an honor to be here. So my goal is to help you understand a little bit about the genetics and how we do it, and I'll tell you at least a couple stories. And the first one is how human genetics is accomplished, specifically with regard to the causes of deafness. I'll talk a little bit about the patterns of transmission of inherited deafness, and I want to then tell you a little bit about the clinical distinction between non-syndromic deafness, deafness all by itself with no other features, and syndromic deafness. So those are the 3 areas I want to cover. So a few comments about hearing loss. It's common. There are about 300 million people in the world that have some hearing loss. About 2 or 3 in 1,000 children are born with a hearing loss. And about half, a little more than half of that, has a hereditary component. And then as we get older, about 50% of 80-year-old individuals have a hearing loss. A part of that is genetic, or at least the risk for that seems to have a genetic component. So you might imagine hearing is very complicated, so many genes are required for normal hearing. Hereditary hearing loss is clinically very diverse, and as you can see from the structure of this inner ear, it's very complicated. It looks like something that the Romans built centuries ago. Today we know that there are about 129 different genes in which variants, mutations, can cause deafness, and that's called genetic heterogeneity. And we also know that many more deafness genes remain to be identified, and that's because we have families that do not have mutations in the known genes, and we're searching for them. And that will be emphasized in a talk that follows me. So, as of May 2019, in a very nice paper that was written by a group at Harvard and with many collaborators, just published in a journal called Genetics and Medicine, all of these genes, which are shown here, have been identified as deafness genes in some paper or papers. However, the evidence in support of all of those genes varies from kind of weak or insufficient to very firm or definitive. So I've put question marks by those where there's weak evidence or insufficient evidence. So over time, this will get sorted out. It isn't an easy process to be absolutely certain that a gene and changes in that gene are associated with the hearing loss in an individual or a family. But for sure, 3 of those deafness genes, which I've indicated by a little puff of smoke, have not only insufficient data, but there's really information now that suggests that they are not deafness genes, that the variants that were found in them were unrelated to the hearing loss that was found in the individuals. And again, over time, with the fullness of time, this will get sorted out. So to study inheritance of humans, there are several steps, and the first 2 are that you need approval from your institution to proceed with a study of human deafness. And that means IRB approval and sort of a contract between yourself and the subject, a meeting of the minds. What am I going to do? What information do I want? And do you agree to provide that information? And then you have to go out and ascertain members of large families, and they need to be cooperative, although some members may not be interested in participating and others will, but there has to be a written meeting of the minds. So I'll come to the other steps in a few moments, but I want to make some more comments about humans and their genetics. And that is, although we look very different from one another, human beings are remarkably similar to one another genetically. About 99% of the coding DNA, the part that's important for making proteins, and those 22 or 23,000 different proteins, they're remarkably similar between humans, but there are enough variants that make us look different. Those variants that make us look different are called the wild type, and they're responsible for the typical form that a species appears in nature. The wild type in an animal, wild types in humans, but in humans and in many other organisms, there's quite a lot of variation, and that's normal. And I want to emphasize that by making some additional points. So natural populations of animals don't have a single wild type. In population, there's a range of variation, and the traits that you see, that's called the phenotype. And wild type variants of a gene are responsible for the typical range of variation that we see, and most organisms have lots of different wild type variants, and those are just called polymorphisms. So a phenotype is taken out of the normal range when there are pathogenic variants or pathogenic mutations. The genetics community now likes to use the word variants instead of mutations. So it's clear that the lower limbs in this individual is not in the normal range. And this was found in a study from Suzanne Leal's lab and my lab, where individuals who were hearing impaired in this family also had this leg abnormality. But we don't know if the same gene is responsible for both. Okay? That's, again, not something that's easy to determine. So the other point that I want to make is the normal function of a gene cannot be deduced directly from the pathology. We know this gene is, some gene is necessary for normal leg development, but exactly what it's doing in leg development or inner ear development or function isn't known simply by looking at the pathology. So the next step is, when you ascertain a family, is to draw a family tree. This is a tree out in the woods, it's dead. You can imagine that at the root of this family are great-grandparents, and then there are children. But in fact, geneticists don't plot it this way. We put the grandparents at the top and the youngest children at the bottom, and we use symbols to represent individuals in the study. So males and females are squares and circles, and when they're darkened in, those are individuals that are affected by whatever trait you're studying. I mean, it could be height or it could be hearing loss. So unaffected individuals that might be carrying the mutation, a recessive mutation, where they fall in the typical normal phenotypic range, have a little dot in the center of the symbol. And then consanguineous marriages, these are marriages between individuals that are related to one another. They're connected by a marriage line which is a double line. If they're unrelated, it's just a single line connecting them. And of course, there can be twins. This is a fraternal twins, or they could be identical twins, in which case they're the same sex. So this is a very simple pedigree, and it shows the inheritance of a recessive trait where the parents are carriers, they have a normal phenotype, and they have, let's say, 4 children, and 1 in 4 carries or has the phenotype. So let's imagine that this is a variant, a mutation on chromosome 5. The parents have 1 normal chromosome, 1 with the variant. They don't have the phenotype. You only know that they are carriers when they have a child with the phenotype that you're studying. So the child inherits 2 mutant alleles. For a dominant, either parent can be affected. In this case, it's the female, and she'll pass on to her children, half of her children, her mutant allele. So that's called an autosomal dominant trait. And I apologize for the nomenclature, I didn't create it, but for recessive deafness genes, they're called DFNB, and then they're given a number, like number 1, I think we're up to about 104 or 105. And for autosomal dominant, they're called DFNA, and they're given a number, like 1, 2, 3, or 4. So this is a hypothetical pedigree of a family segregating a recessive trait. And you notice that there are a number of individuals in the last generation, and they are affected, and there are carriers. And I've left out a single individual that should be a carrier in this pedigree. You might look to see if you can find it. But sometimes when an individual won't participate, or the parents, you don't know who is the carrier. Okay, so to remind you, humans have 46 chromosomes. We have 23 pairs. We get 23 from dad and 23 from mom. We have 46. It's just that when I was in high school, when I entered high school, humans had 48 chromosomes. By the time I finished, every human on the planet had 46 chromosomes, because it was just hard to count chromosomes when I was in high school. But in addition to having 46 chromosomes, males have 1 X and 1 Y. They get their Y from their dad, their X from their mom. But we also have another chromosome in our mitochondria, and I'm going to come back to that in a moment. So the pattern of transmission of inherited human deafness can be autosomal recessive on one of those 22 chromosomes, or dominant on one of the 22. And it's about 59% so far that are recessive variants that cause deafness, and about 36% that cause dominantly inherited deafness. But there is this other 5%, and 4 of it is the result of mutations that are on the X chromosome that are associated with hearing loss. So that's the X chromosome with the little green egg-shaped circle around it. So what about the other 1%? Well, the other 1% are mutations in the mitochondrial chromosome, and I'll tell you what that is in a moment. So that's circled. It's a little chromosome, and in a cell, and this is a single cell with the outer cell membrane and the nucleus where the chromosomes are located, but in all of those little mitochondria where there are yellow areas and yellow arrows, there's also a mitochondrial chromosome, and variants of the mitochondrial genome also can cause hearing loss. So that mitochondria in that oocyte, which will eventually develop into a human, those mitochondria are all contributed by mom. They're all in the egg. The sperm does not contribute a mitochondria. So mitochondrial inheritance is maternal. It has a maternal lineage since the male doesn't contribute. So how are genes organized on chromosomes? And I like to think of genes as houses, and I like to think of the parts of genes as rooms in a house. So genes are located along chromosomes, but they're not like these brownstones where they're one right next to another. There are genes that are right next to each other, but most genes are separated by a lot of space. A lot of DNA separates genes from one another. So like this country house. And not only are there genes that are separated from one another, but there are big genes and there are tiny genes, just like this tiny house. And if you imagine again that houses are genes, only about 1 or 2% of our genome are genes. The rest of it is the space between genes and the space between parts of genes, like those rooms in a house. So I'm going to tell you about a big gene and a little gene. So before I get to that, I want to just tell you the remaining steps that you need to do to study these kinds of genes. So we've ascertained a large family. This is a nice family. We acquire audiological data. Usually for my lab, we don't acquire much of any other clinical data at the time. We then try to determine where the gene is located on which one of those 23 chromosomes, or on the mitochondrial chromosome the gene is located, and I'm not going to bore you with the steps to do that. So once we've identified the gene, we try to study the function of the gene, and you can't study the function of that gene very easily in humans. So you engineer animal models to test your hypothesis about what the function of that gene might be. And then you do functional studies. You try to figure out what is the role of that gene in a cell in the inner ear. And then you begin thinking about therapies, small molecule therapies, gene therapies. But you need animal models. And the two popular animal models are the mouse and zebrafish, although fruit flies are also good models to study the functions of genes because they're much less expensive and you can raise thousands and thousands of them. So you create animal models if there isn't one already available, and you study the phenotype of those variants of that same human gene, but in these animal models. And you ask, well, what's the pathogenic mechanism? And the same question, or a similar question, is what's the normal function of that gene? So step 9 is that when we study the function of these genes in these animal models, we almost always discover something unique or interesting, or we discover another phenotype that seems to co-segregate with the deafness. So we go back to the family and we ask for more clinical data. So if in our mouse model and our human we don't find anything else co-segregating with the deafness is said to be non-syndromic. So if there's no accompanying disorders, the deafness is non-syndromic. If the deafness is accompanied by another disorder and we can causally connect it in our animal model to the variants that we see in humans, but we're studying the animal model, the deafness is said to be syndromic. So when there are other additional abnormalities of other tissues and organs due to that genetic change. And there are lots of syndromes that involve humans, but here are 4 that I've listed. One is Usher syndrome, which is deafness and loss of vision due to retinitis pigmentosa. Alport syndrome, which is deafness and kidney disorder. Perot syndrome is deafness and ovarian dysgenesis, or premature ovarian failure, whereas the males seem to be fine in terms of fertility. And a new syndrome, which we published a year ago, which we call HIMSS, which stands for inherited hearing loss and infertility in males. So it's just the reciprocal of Perot syndrome, where there's deafness and infertility in males, but the females are fertile. That's true for the mouse model, it's true for the human families. So there are about 142 different deafness genes that have been reported to cause human loss of hearing. But if you go to the online Mendelian Inheritance of Man, which is supported by Johns Hopkins University, you'll find that there are about 500 syndromes where deafness is one feature. And we really only have identified the genes for about a quarter of those, or maybe even just a fifth. So what you'll notice in the pie chart, however, is that 57 plus 106 does not equal 129. It's more than 129. And since I can do addition, the question is, how come that number is larger than the 129? And the answer to that is that different variants, different mutations in the same gene can cause either syndromic deafness or non-syndromic deafness. Some variants cause the syndrome. Other variants, which aren't as disabling to the gene, can cause just non-syndromic deafness. So there are about 19 human deafness genes where different variants can either cause syndromic deafness or, if it's less disabling, non-syndromic deafness. Okay, so in the remaining couple minutes, I just want to tell you about a tiny gene and a big gene. So GJB2 is the gene that encodes connexin 26, or CX26, or some people call it, actually it's DFNB1. So mutations in GJB2 encoding connexins are major contributors to inherited deafness in many, but not all, ethnic groups. And this little tiny gene, as of a couple days ago, there are 428 different pathogenic variants that can disrupt this gene. And again, it's a tiny gene, and its function is to allow for communication between adjacent cells. So that little red dot is in one cell, and it can go through a connexin channel and end up in the adjacent cells. So cells talk to one another by moving molecules between them. They're not just isolated and not communicating with one another. So some mutations of JGB2 are exceedingly rare. You find them in just one or two families, and others are very common. So I won't bore you with the details of what these names mean for these variants, but the 35del-G mutation is very common in Southern Europeans in the United States, and about 3% of individuals in Europe and USA carry a mutation in that gene. They're unaffected, but they're carriers, and if they mate or marry an individual who's a carrier, about 1 in 4 of their children will have both mutations and have a hearing loss. The 167 del-T mutation is common in the Ashkenazi Jews. Ashkenazi Jews don't have more hearing loss than any other group, but one mutation is responsible for at least 60% of the non-syndromic deafness. And 4% carry this mutation, and it's because of a bottleneck in the history of the Ashkenazi Jews. And in the Japanese, it's another mutation, 235 del-C, and 1% carry this mutation. So when you have a subject in your clinic and they indicate they have an Ashkenazi Jewish background and they have hearing loss, that's the mutation you look for first. Okay, so now for a big gene. And this one we discovered in a Balinese village called Bengala about 25 years ago. There are 2,200 people in the village. We didn't make this movie, but you can go to YouTube and you can see it's a very nice video about the village of the deaf. And it is true that almost everyone in the village knows sign language. So when you see people signing to one another, you don't know whether they're hearing or they're hearing impaired. The village is located in the north of Bali, very close to a city called Singaraja. And that's the pedigree. In fact, we have a pedigree of all 2,200 in the village. But this is a portion of that entire village. And there were, at that time, 48 deaf individuals. We mapped the deafness gene to chromosome 17. The location on that chromosome was called DFNB3. And it turned out to be a very big gene. So it's a big house with 67 rooms instead of one like GJB2. And I know you can't read it, but these are all the mutations as of a couple of weeks ago. And the one in the village is just one in those in particular. It has been slow to study in individuals with hearing impairment because it's such a large gene, and it used to take a lot of work. But depending on the population, it's either the third or the sixth most common gene. And although we were criticized for going and studying deafness in a Balinese village, and so what? What would be the impact of that? Mutations in this gene have been found in individuals with hearing impairment worldwide. And it raises the importance of having collaborations worldwide and working with scientists worldwide and studying communities worldwide. So to summarize, there are many different deafness genes in the human genome. We have not identified all of them by any means. The strength of the evidence for those deafness genes varies from limited or weak to definitive. The way to study them is with animal models, and they're essential for studying the functions of these genes. And many rare deafness genes remain to be identified, and even if they're only found in a single family, when you make an animal model, you'll learn something important about the auditory system, which is very complex. And then I want to stress again that international research collaborations have been instrumental to the success in identifying genes for hearing and other sensory disorders, many different sensory disorders. And we should continue to emphasize the importance of working with other researchers around the world. So if you have questions when we're finished, I'll be happy to answer them, and I'll be here all day, and you're welcome to come up and talk with me about anything that you're interested in or any point that I've made. And I just want to end by acknowledging Andy Griffith, who I've been studying hereditary deafness for about 20 years. He came up through my lab, and he's my boss now. Ina Belyansova, who's a staff scientist in my lab. And then in the lower, your lower left, is John Hinnant, who's a medical anthropologist. You can't study other communities in the world without having a medical anthropologist working with you. It's absolutely critical. When you ask somebody who's your mother, it's got to be clear that you want to know the birth mother, not the person who raised you, for example. And then my close friend and research colleague in Pakistan, Sheikh Riazuddin. So thank you very much.